Bismillahirrahmanirrahim is part 2 of chapter number 5, resting membrane potential of neurons. How the resting membrane potential develops in the neuron. Now we will discuss in more detail. For example, if this is the sodium potassium channel, if this is a sodium potassium pump, this sodium potassium pump carries 2 potassium ions to the inside of cell and 3 sodium ions to the outside of cells. So in this way, the potassium pump, which we have studied in chapter number 3, now we will discuss the application of this potassium pump. Now, what is the most important functions of the potassium? The potassium is important in every cell of the body, but for the neurons, the most important, for the function of neuron, the most important thing is the sodium potassium pump. So the sodium potassium pump carries 3 sodium to the outside and carries 2 potassium to the inside and generate a concentration difference. Now, when this potassium pump is activated, then it's produce the concentration difference of 142 milliequivalents of sodiums accumulate milliequivalents per liter accumulate in the extracellular fluid. The potassium inside accumulation of the potassium is 140 milliequivalents per liter. The sodium which remains inside the cell is 14 milliequivalents of the sodium. Only this amount remains inside the cell, and outside the cell only 4 milliequivalents of potassium remains outside the cells. So with the help of this sodium potassium pump, with the help of this sodium potassium pump, 142 milliequivalents of sodium accumulates outside and 140 milliequivalents of potassium accumulates inside the cell, and 14 milliequivalents of sodium, only 14 milliequivalents of sodium remains inside, and 4 milliequivalents of potassium remains outside of the cells. So in this way, this sodium potassium pump is responsible for these concentration differences in accumulation. Now we will need their ratio for the to put in the Goldman equation and to calculate the respective membrane potential. So the ratio for the sodium inside divided by sodium outside. So if we divide in the inside, sodium is only 14, and if we divide this by the sodium which is the outside of the cell is 142, then we will get only a value of 0.1. This is the ratio of the sodium. This is the ratio of the sodium inside and to the sodium which is outside is 0.1. Now we will calculate the ratio for the potassium. The ratio for the potassium is potassium inside divided by potassium outside. Potassium on the inside is 140 milliequivalents and on the outside it is 4 milliequivalents. So we got a ratio of 35 ratio 1. So we got this. Now this is the sodium. This is the ratio for the sodium and this is the ratio for the potassium. Now we will need these ratio in the upcoming discussion. So this is the contribution of the potassium pump in generating the ionic concentration difference across the membrane. Now we will discuss. It is done leakage of potassium. You know, across this cell membrane is present a type of channel which are called the leaky channels. These are the leaky channels for potassium. Leaky channels for the potassium. This leaky channel is responsible. It is more permeable for potassium as compared to the sodium. This leaky channel, which is also called potassium channel or potassium leaky channel, are tandem pore domains. This is 100 times more permeable to the potassium as compared to the sodium. So this channel is also responsible for the resting membrane potential to develop. Now, this was one thing which was responsible for resting membrane. This is the second which is responsible for the resting membrane. Now we will discuss the third, which is important. The third is the original potassium channels, which we have discussed in chapter number 3. The third thing is the potassium channels. These potassium channels has its selectivity filter, which we have discussed, which is lined by the carbonyl oxygen, and they only allow the potassium ion to pass through it. So these potassium channel has one gate which remain closed, but if the membrane potential rises from -70 to 0 for small neurons, or if it is large for the small neurons. For the large neurons, if it rises to +35, large neurons. This is the potassium channel, which has a selectivity filter lined by the carbonyl oxygen, which allow only the potassium channel to pass through it. Now, this is the potassium gate. This is the gate of the potassium channel, and this gate remain closed. When the sodium channel carries positive charges inside, then when the resting membrane potential rises to 0 or to +35. For the small neuron, if it rises to the 0. For the large neuron, if the resting membrane potential rises to +35, then what will happen? The gate of potassium will open, and the role of this potassium gate is to regain its original polarity again, which was disturbed by the sodium. The sodium disturbed the electrical polarity inside the cell membrane and causes the depolarization. Now the gate of the potassium will regain its polarity and will open this gate if the resting membrane potential rises to 0 +35, and this gate will open and it will allow the potassium channel to pass to the outside of the cell and regain its polarity again. It will open like in this manner, and it will allow the potassium ions to pass to the outside. But this potassium channel is highly specific for the potassium and allow the potassium. This potassium channel is highly specific for the potassium and allow the potassium to pass to the outside of the cell. And we have discussed the potassium leaky channel, which is responsible for the potassium mainly, but it is also permeable to sodium. But the permeability to sodium is 100 times less than to potassium. So the leaky channel is more permeable to potassium. So we have discussed the three routes how the cell regain its normal original polarity and electronegativity inside. First was the sodium potassium pump, second was the leaky channel, and the third is now the depolarization produced by the sodium. Now this depolarization is terminated by the potassium channels. Now we will discuss the contribution of each of these phenomena, that how much each of these phenomena has contributed to the resting membrane potential. For example, now we will take a look to the contribution of these ions in the generation of the resting membrane potential. For example, we have previously we have used the formula that the EMF is equal to the formula which has been used for the Nernstequation potential. So log divided by Z multiplied by concentration inside divided by outside. So we will calculate the potential of each of these ions, that how much each of these ions is contributing. So, for example, and plus or minus 60, it is. Now we will apply the formula of the electromotive force or Nernst potential, that how much of these each of these ions is contributing to the resting membrane potential. Now, for example, potassium, which is 1 4 on the inside it is 140, on the outside it is 4. We got a ratio of 35. Now the log plus minus 60 log 35. The logarithm of 35 is 1.54. Now we have discussed that if the charge which is coming out of the cell, the potential charge will be equal to negative. If the charge is coming out of the cell is negative, then the sign will be positive, and vice versa. So for the potassium we will take the sign minus 60. So it will, when we multiply 1.54 by minus 60, minus 94 comes the answer. Millivolts. This is the contribution of potassium in the generation of the resting membrane potential. Now we will use this formula, this the electromotive force or the Nernst potential formula for the sodium. So for the sodium, for example, sodium on the inside is 14, on the outside sodium is 142 milliequivalents per liter. Their ratio is 0.1. So when we take their logarithm, please calculate the logarithm, guys. It's difficult to calculate 0.1 logarithm. So the answer comes plus 61 millivolts. Why the sign for the sodium comes positive, but for the potassium it has come negative? Because the potassium which is diffusing outside and the sodium is diffusing inside. That's why due to their direction, their charge has been changed. Now this is the potential. This is the potential of potassium, and this is the potential which is contributed by the sodium. So this is the potential difference, which is minus 86 millivolts. How it comes? For example, take a look over this that the Goldman equation is used to calculate the diffusion potential when the membrane is permeable to several different ions. So if a particular membrane is permeable to several different ions, we will use the Goldman equation, and through the help of this Goldman equation we can calculate the potential difference of minus 86 millivolts. Now this is the contribution of the sodium and potassium. So we have discussed that through this the potassium is leaking and the sodium is also leaking, and through this potassium channel the potassium is leaking. So the contribution of sodium through the potassium leaky channel, the contribution of potassium through the leaky, and the contribution of potassium through the potassium channel has discussed there, how they contribute to the generation of resting membrane potential. Now how much the potassium pump contribute to the resting membrane potential? It has only a contribution of minus 4 millivolts. So the sodium potassium pump contributes only minus 4 millivolts. And if all these three things, the potassium leaky channels through which the sodium is also leaking, and the potassium channels and the sodium potassium pump, if all these three factors are operating at the same rate, the resting membrane potential will be minus 90 millivolts. So this was about the resting membrane potential. Now we will discuss the neuron action potential, how the action potential develops. Now neuron action potential. When any kind of stimulus, whether it is chemical, it is mechanical, or it is electrical, when it disturbs the surrounding of a neuron, because of these disturbances, the inside of the cells becomes positive, and this positivity rises to such an high value and adequate value that it can depolarize the neuron. Then an action potential develops. For example, if the large neuron has the threshold for the action potential of minus 55 millivolts, if any change occurs around the neuron, It disturbs the inside positivity of the neurons from minus 70 millivolts to minus 55 millivolts. This disturbance in this threshold potential which has developed, it will causes the opening of the sodium channel and will causes the depolarization of the membrane, and in this way the action potential develops and the impulses travel along the neurons. So so the resting stage, we have discussed how the resting membrane potential develops. So the resting membrane potential develop when the potassium is coming to the inside and the sodium is coming to the outside of the cell, and the potassium channels and the potassium leaky channels are also working, so they bring the negativity of the inside of the cell to a normal resting value of minus 70 millivolts. This is the resting state. Now the depolarization, which I have, that any disturbance which disturbs the resting membrane potential and carries the resting membrane potential of minus 70 millivolts to a threshold value which is minus 55 millivolts for the large neurons. When this resting membrane potential rises up to minus 55 millivolts, then what happen? Then this minus 55 millivolts causes the opening of the sodium channels, and the sodium in turn diffuse to the inside of the membrane and carries out the depolarization, and the action potential develops in this manner. The repolarization stage, again within a few or 10,000 of a second, again the repolarizing operation starts, and the sodium potassium pump activates the potassium channel activate, and the sodium potassium leaky channel also activate. So these activation leads to the development of the repolarizing stage, again with the negativity inside and the positivity outside of the cell membrane. Now the voltage gated sodium and potassium channels. So I have discussed this in the previous chapter, so we will take a look through this. This is, for example, we have discussed two types of gates. The one was the voltage gated and another was the ligand or chemical gated. So we will focus on the voltage gated. This is the sodium channel, and this is the outside gate is the activation gate, and the inside gate is inactivation gate. Well, for example, any disturbance, whether it is chemical, mechanical, electrical, any kind of stimulus which disturbs this the electronegativity of the inside of the membrane, any disturbance which rises this negativity of minus 70 to minus 55 millivolts. So this is the threshold for the opening of this activation gate. This minus 50 millivolts will open this the activation gate of sodium, and the sodium ion will diffuse to the inside and will develop the depolarization and action potential. And if this depolarization again rises to a certain value which is plus 35 in case of large neuron, then the sodium inactivation gate will activate it, and this inactivation gate will closes. This inactivation gate will closes the sodium channel, and will prevent the further diffusion of the sodium onto the inside. Now at this voltage, the inactivation gate of the potassium has been closed. Of the sodium has been closed. Now the gate of the potassium will open. The gate of the potassium will open, and this will regain its original polarity and negativity by removing the positivity ions, the positive potassium ions from the inside of the cell, and regain its polarity. But the time at which this inactivation gate of sodium start closing, this gate of the potassium start opening. This is for example, this is the potassium channel. When the sodium channel start closing, then the potassium channel start opening. And in this way, for most of the time for which the depolarization was occurring, at that point the potassium gate was closed. But when this activation gate is closed, then the potassium gate start opening, and in this way they remove the extra positive ions from the inside pool of the cell and regain its original polarity in resting state again. So this was about the voltage gated sodium and potassium channel. This was about the activation and inactivation gates. The voltage clamp method. The voltage clamp method is used for the effect of the voltage on the opening and closing of the channel. And what is the effect of the voltage on the behavior of different types of currents? So the voltage clamp method is used for this aim. Now we will first discuss the arrangement of the apparatus of the voltage clamp method. For example, This is the giant squid neuron. This is a saline solution. This the giant squid neuron is dipped in the saline solution. This is the reference electrode, and this is the recording electrode. The reference electrode and the recording electrode is connected to the amplifier. This is the current electrode. The current electrode is connected to the ampere meter, and this is in turn connected with the comparator or feedback amplifier. And this is the area from which we can desirably change the voltage according to our desire. We can change the voltage from this area. So this is the arrangement of the voltage climb method. Now we will discuss in more detail the principle of the voltage climb method. Now let's discuss the principle of the voltage climb method. This is the independent electrode. This is the recording electrode. We have discussed the independent electrode and the recording electrode is used for the measuring the membrane potential. The ampere meter is used for the measuring of the current which is passing through the current electrode. Now, if the amplifier is showing minus 70 millivolt voltage, so it means that inside the axon the voltage is minus 70 millivolt. And now if we change the voltage desirably to minus 70 millivolt, if we change the command voltage desirably to minus 70 millivolt and the amplifier voltage is also minus 70, the comparator will compare these two voltage and it will produce no current and no signal. So no deflection will be shown by the ampere meter and no current will be injected to the axon. So when the membrane potential is equal to the command potential, no current will be generated. The current will be zero. But if we, for example, raise this command potential to minus 30, if we raise the command voltage from minus 70 to minus 30, then this comparator will compare these two voltages and will produce the signal, and the current will pass through the ampere meter and the deflection will be shown by the ampere meter, and this current will go through the current electrode to inject into the axons. When this positive current comes through this current electrode, then it will raise this potential from minus 70 to minus 30 because of the injection of these positive current. Now in order to equalize the potential on the both side, the sodium channel, the activation gate, this is the sodium channel. So the activation gate of the sodium channel will open and it will permit the sodium ions to flow. So the sodium channel will permit the sodium ion to flow to the inside of the axon and to decrease the outside positivity to minus 30. So when the voltage inside the axon and outside the axon is equalized, again the ampere meter will show no current, and in this way we can change the current through the current electrode, and we can study the behavior of different type of channels. This was the principle of the voltage climb method. So if we further increase this voltage to minus 20, minus 10, etc., the positive current will rise this internal voltage of the axon, and accordingly the outside potential will also change, and these two potential will equalize each other, and in this way we can study the conduction of current through different types of channels. So this was the basic principle of the voltage climb method. Now it's done. Part 2 of chapter 5 has been done. Now guys, please subscribe my channel, like, subscribe and comment. I will be very thankful to you.